Viral pathogenesis describes the processes by which viral infections cause diseases and involves virus-host interactions at the cellular and systemic level that determine whether a virus will cause a disease, what form the disease takes, and how severe the disease will be. Though the pathogenesis of each virus is unique, there are several common points in the virus life cycle that are shared between all pathogenic viruses, and by considering these common aspects of the virus-induced disease process, we can explore some general concepts in viral pathogenesis while illustrating some of the specific processes that shape disease outcomes. Okay, when a virus infects a host, there are three basic problems that it faces. Of course, it has to be able to reproduce within the host's cells, which requires entering the cell, copying genetic information, making messenger RNA so that viral proteins can be produced. The next step is, how does the virus spread from one individual to another? If the virus can't move on to another host, another individual, then it's at a dead end. And lastly, how does the virus evade host defenses? The diseases that viruses cause are the consequences, usually unintended, of the way each virus solves the problems of reproduction, spread, and evasion. And that's part of what we're going to look at in this module. This diagram illustrates the classic iceberg concept of infection, dividing the seen or discernible effects of a disease process from the effects that are unseen. We can look at this at the cellular level or from the host level. The disease process begins at a point of inoculation, reaching the prodromal or earliest onset of symptoms, and eventually becoming a full-blown observable clinical disease, where it reaches the tip of the iceberg. Those patients that are visibly infected with an organism represent just the tip of the iceberg of patients that are colonized or infected. Just because a patient is not showing signs of infection does not mean that he does not carry organisms that could be transferred to another patient if proper hygiene and other infection control precautions are not taken. And in fact, we're seeing just exactly this problem with COVID-19. We find that asymptomatic people are capable of transmitting the disease. The first line of defense against non-self pathogens is the innate or non-specific immune response. The innate immune response consists of physical, chemical, and cellular defenses against pathogens. The main purpose of the innate immune response is to immediately prevent the spread and movement of foreign pathogens throughout the body. The second line of defense against non-self pathogens is called the adaptive immune response. Adaptive immunity is also referred to as acquired immunity or specific immunity, and is only found in vertebrates. The adaptive immune response is specific to the pathogen presented. It is also meant to attack non-self pathogens, but can sometimes make errors and attack itself. When this happens, autoimmune diseases can develop, such as rheumatoid arthritis. While the innate immune response is immediate, the adaptive immune response is not. However, the effect of the adaptive immune response is long-lasting, highly specific, and is sustained by long-term memory T cells. Physical barriers play an important role in preventing microbes from reaching tissues that are susceptible to infection. At the cellular level, barriers consist of cells that are tightly joined to prevent invaders from crossing through to deeper tissue. For example, all surfaces of the body, internal and external, are covered with epithelial cells. One of the body's most important physical barriers is the skin barrier, which is composed of 3 layers of closely packed cells. The thin upper layer is called the epidermis. A second, thicker layer, called the dermis, contains hair follicles, sweat glands, nerves, and blood vessels. A layer of fatty tissue called the hypodermis lies beneath the dermis and contains blood and lymph vessels. The topmost layer of the skin, the epidermis, consists of cells that are packed with keratin. Only the basal layer of cells divide, and as new cells form, old cells are pushed to the surface where they become flattened and die. These dead cells remain as a tightly connected, dense layer of protein-filled cell husks on the surface of the skin. The keratin makes the skin's surface mechanically tough and resistant to degradation by bacterial enzymes. Fatty acids on the skin's surface create a dry, salty, and acidic environment that inhibits the growth of some microbes and is highly resistant to breakdown by bacterial enzymes. In addition, the dead cells of the epidermis are frequently shed, along with any microbes that might be clinging to them. Shed skin cells are continually replaced with new cells from below, providing a new barrier that will soon be shed in the same way. Infections can occur when the skin barrier is compromised or broken. For instance, a wound can serve as a point of entry for opportunistic pathogens, which can infect the skin tissue surrounding the wound and possibly spread to deeper tissues. The mucous membranes lining the nose, mouth, lungs, and urinary and digestive tracts provide another nonspecific barrier against potential pathogens. Mucous membranes consist of a layer of epithelial cells bound by tight junctions. The epithelial cells secrete a moist, sticky substance called mucus, which covers and protects the more fragile cell layers beneath it and traps debris and particulate matter, including microbes. Mucus secretions also contain antimicrobial peptides. In many regions of the body, mechanical actions serve to flush mucus, along with trapped or dead microbes, out of the body or away from potential sites of infection. For example, in the respiratory system, inhalation can bring microbes, dust, mold spores, and other small airborne debris into the body. This debris becomes trapped in the mucus lining the respiratory tract, a layer known as the mucociliary blanket. The epithelial cells lining the upper parts of the respiratory tract are called ciliated epithelial cells because they have hair-like appendages known as cilia. Movement of the cilia propels debris-laden mucus out and away from the lungs. The expelled mucus is then swallowed and destroyed in the stomach, or coughed up or sneezed out. This system of removal is often called the mucociliary escalator. The mucociliary escalator is such an effective barrier to microbes that the lungs, the lowermost and most sensitive portion of the respiratory tract, were long considered to be a sterile environment in healthy individuals. Only recently has research suggested that healthy lungs may have a small, normal microbiota. Disruption of the mucociliary escalator by the damaging effects of smoking or diseases such as cystic fibrosis can lead to increased colonization of bacteria in the lower respiratory tract and frequent infections, which highlights the importance of this physical barrier to host defenses. Like the respiratory tract, the digestive tract is a portal of entry through which microbes enter the body, and the mucous membranes lining the digestive tract provide a nonspecific physical barrier against ingested microbes. The intestinal tract is lined with epithelial cells interspersed with mucus-secreting goblet cells. This mucus mixes with material received from the stomach, trapping foodborne microbes and debris. The mechanical action of peristalsis, a series of muscular contractions in the digestive tract, moves the sloughed mucus and other material through the intestines, rectum, and anus, excreting the material in the feces. The epithelial cells lining the urogenital tract, blood vessels, lymphatic vessels, and other tissues are known as endothelia. These tightly packed cells provide a particularly effective frontline barrier against invaders. The epithelia of the blood-brain barrier, for example, protect the central nervous system, or CNS, which consists of the brain and the spinal cord. The CNS is one of the most sensitive and important areas of the body, as microbial infection of the CNS can quickly lead to serious and often fatal inflammation. The cell junctions in the blood vessels traveling through the CNS are some of the tightest and toughest in the body, preventing any transient microbes in the bloodstream from entering the CNS. This keeps the cerebrospinal fluid that surrounds and bathes the brain and spinal cord sterile under normal conditions. The innate immune system is composed of a large number of elements that attempt to control infection by pathogens, but this system is independent of the identity of the particular pathogen. The innate system uses cytokines, which are soluble proteins released from infected cells such as interferon, local sentinel cells like dendritic cells and macrophages, serum proteins such as complement, and cytolytic lymphocytes. It relies on unusual receptors that reside on the surfaces of sentinel cells, the so-called pattern recognition receptors. These receptors detect non-self targets or missing self or altered self, and then unleash molecules to kill the invader and the cells harboring it. This system is activated quickly and can begin functioning within minutes to hours of infection. In contrast, the adaptive response takes much longer, usually days or even weeks. The innate immune response is rapid, but also must be transient because its continued activity can be damaging to the host. Things to remember about the innate immune response is that they are present from birth, they're nonspecific, and they do not become more efficient over time. The innate immune system is extremely important. First of all, it's the only defense available for the first few days after a viral infection. It takes antibodies, which are part of the adaptive immune response, much longer to develop. It is the only system that can discern the general nature of the invader. And it's the only system that can inform the adaptive immune system when infection reaches a dangerous threshold. So the innate immune system will communicate with the adaptive system to tip it off as to what kind of invader it might be, and also it lets the adaptive immune system know that this infection is getting out of control. Further help is necessary. Phagocytic and natural killer cells are important components of the innate immune system. Damaged cells release chemical signals that attract neutrophils from the blood. They enter the infected tissue and engulf and destroy microbes. 60 to 70% of our white blood cells are neutrophils. Monocytes move to tissues and develop into macrophages. They engulf microbes using pseudopodia and bring it in a vacuole. They can use nitric oxide or lysosomal enzymes to kill. Some macrophages migrate and reside permanently in certain tissues, especially the lymphatics. Foreign agents that enter the blood encounter macrophages as they become trapped in the net-like structure of the spleen, for instance. Those in tissue fluid flow into the lymph and are filtered by the lymph nodes. Eosinophils are primarily a defense against larger parasites, such as blood flukes and nematode worms, and so they're really not of particular importance when we're considering viruses. Natural killer, or NK cells, are cytolytic cells that kill by an antigen-independent mechanism. It's important for the control of many virus infections. For instance, they've shown that ablation of NK cells leads to more serious disease in animals. NK activity increases very early after infection by a virus, within the first 2 to 3 days, and then the number declines. MHC, or major histocompatibility complex surface glycoproteins, classes 1 and class 2, mark body cells as self. NK cells kill cells that do not express MHC or express it only in low amounts. Many viruses downregulate the production of MHC in infected cells to escape immune surveillance. NK cells evolved to rid the body of cells that would normally escape the immune system, such as virus-infected cells or tumor cells. The inflammatory response is an important component of the innate immune system. In an injured area, precapillary arterioles dilate and postcapillary arterioles constrict, increasing the local blood supply. That's why in an area that's infected, you get redness, and when you touch it, it feels hot. The blood-engorged capillaries leak fluid into neighboring tissues, causing edema or swelling. This response is initiated by chemical signals. For instance, histamine triggers both dilation and increased permeability of nearby capillaries. Prostaglandins are also released by damaged cells and further promote blood flow to the site of the injury. Enhanced blood flow and capillary permeability aid in delivering clotting elements to an injury. Blood clotting marks the start of repair and prevents the spread of microbes to other parts of the body. Increased blood flow and capillary permeability also enhance migration of phagocytic cells from blood to injured tissue. Neutrophils are followed by macrophages. Macrophages will engulf pathogens, but also cleans up the damaged tissue cells and the remains of neutrophils. Pus that forms at the site of an infection consists mostly of dead phagocytic cells and the fluid that leaked from the capillaries during the inflammatory response. The body can also mount a systemic nonspecific response to severe tissue injury or infection. Chemotactic chemicals stimulate the release of more neutrophils from blood marrow. The number of leukocytes in the blood can increase several-fold within a few hours of infection. Pyrogens are released by leukocytes and set the body's thermostat higher. Therefore, you get a fever. Fever can facilitate phagocytosis and speed up body reactions. So within certain limits, fever is actually a good thing in response to an infection. We also have antimicrobial proteins that are part of the innate immune system. We have the complement system, we have apoptosis, and we have interferon. So we'll talk about each of these in more detail separately. So first, let's talk about the complement system. Although the complement system has traditionally been considered part of the innate immune system, research in recent decades has revealed that complement is able to activate cells involved in both the adaptive and the innate immune system. Complement triggers and modulates a variety of immune activities and acts as a linker between the two branches of the immune response. In addition, the complement system maintains cell homeostasis by eliminating cellular debris and immune complexes. So it actually acts as a bridge between the innate and adaptive immune systems. But we'll talk more about its role right now as part of the innate system. So the complement system is composed in part of more than 20 soluble proteins that circulate in the blood. These proteins are activated through a proteolytic cascade to produce effector molecules that aid in the control of infection. It plays a role, as I said, in both the innate and the adaptive immune systems. Complement proteins are made primarily by the liver. The most abundant one is C3. C3 molecules are constantly being broken down into two smaller proteins spontaneously. One product is C3b, which can bind to chemical groups commonly found on the surfaces of viruses. C3b can catalyze a series of reactions that result in the cutting of many more C3 molecules, a chain reaction that results in the deposition of large numbers of C3b fragments on the surface of the virus. Macrophages have receptors that bind to C3b. The virus has had its surface, we call it decorated or opsonized, with C3b, making them preferred targets for macrophages. C3b can also bind to the surfaces of envelope viruses. The C3b proteins can initiate a biochemical reaction that results in the formation of membrane attack complexes, which can create holes in the virus envelope, and then it's all over for the virus particle. C3a, the other piece, doesn't bind virus, but it can recruit macrophages and neutrophils. The system can only get at viruses that are outside of the cell. Apoptosis is programmed cell death. It's really the defense of last resort for an infected cell. It's a cell suicide pathway in which mitochondria cease to function, chromatin in the nucleus condenses, nuclear DNA is degraded, and the cell fragments into smaller membrane-bound vesicles that are phagocytosed by neighboring cells. It serves to eliminate cells that are no longer needed, cells that are damaged, cells that are infected, or cells with a deregulated cell cycle. Those would be potential tumor cells. Mitochondria are important players in apoptosis. A decrease in the potential across the mitochondrial membrane makes the cell more sensitive to apoptosis. Decreased potential is accompanied by synthesis of reactive oxygen and the release of cytochrome C into the cytoplasm. These are pro-apoptotic events. Regulation of apoptosis is complicated and beyond the scope of this course. However, it's important to be aware that virus infections perturb cellular biochemistry and can trigger an apoptotic response. Apoptosis involves a cell degrading itself from within, digesting its DNA, and signaling phagocytes to clear up the debris. Necrosis does not. Necrosis results when a cell dies unexpectedly, for example, from oxygen starvation or the effects of toxins. While the intracellular events during necrosis superficially resemble those of apoptosis, there's not the same pre-programmed clearing up procedure. As a result, inflammation and enhanced toxicity can result with the potential for destroying large areas of tissue. So apoptosis can be protective. Necrosis tends to be a more damaging kind of result. Interferons are cytokines that are part of your natural defenses. Cytokines are small, soluble proteins made by one cell that can affect the behavior of other cells. In other words, they're signaling molecules. Interferons got their name because they interfere with viruses and keep them from multiplying. Almost every cell in your body makes interferons. There are three main types: interferon alpha, interferon beta, and interferon gamma. Cells that have been infected with viruses give off interferon alpha and interferon beta as a warning signal to your immune system. That triggers white blood cells to release interferon gamma to fight the invader. Interferons work in a few different ways. They alert your immune system so it can go after the virus or cancer. They help your immune system recognize the virus or cancer. They tell immune cells to attack. They stop viruses and cancer cells from growing and dividing, and they help healthy cells fight infection. Here in this slide, you can see the effects of type 1 interferon levels on the response of mice to dengue virus infection. Dengue virus is a mosquito-borne flavivirus, and symptoms of infection range from asymptomatic to the severe dengue hemorrhagic fever. High viral loads correlate with disease severity, and both type 1 and type 2 interferons are crucial for controlling viral replication. Now, on this slide, if you look over on the left side, you can see there's WT, STAT1, STAT2, and all that. These are different strains of mice. WT is wild type. The other three types have altered interferon responses, and you can see that clearly in that graph. The graph is showing the amount of interferon alpha produced in the hours post-infection, and you can see very clearly that the wild-type mice produce more interferon alpha than the other three types of mice. Well, if we test the production of dengue in these different types of mice, that's what you see on the right-hand side. And across the bottom of the graph, you just have hours after infection with dengue. And here we're looking at the amount of dengue produced, so the log dengue produced. And again, it's those different strains of mice. The black bars are the wild type. And if you look at 6 hours post-infection, all of the mice have a similar response to dengue. But as you move out to 12 hours, 18 hours, and so on, you can see that the wild-type mice show a much lower level of dengue virus production compared to the ones that have an altered interferon response. The host innate immune response mediated by type 1 interferon and the resulting upregulation of hundreds of interferon-stimulated genes, or ISGs, provide an immediate barrier to virus infection. IFNs induce the production of several known antiviral proteins, and these include PKR, MHC. Until the advent of DNA microarrays, it was thought that there were perhaps 30 to 40 ISGs, a small number of which were thought to have antiviral proteins. Now we know that there are hundreds of IFN-regulated genes, many of which are repressed or downregulated by IFN, and you can see an example of one of those arrays on the slide. Okay, now we want to talk about the biological activities of IFN. IFN leads to increased production of MHC molecules, thus leading to the activation of natural killer cells, monocytes, macrophages, and CTLs. Interferons are pyrogenic. They can cause high fever. Now remember, I said that to a certain extent, fevers can be useful when you have an infection. Fever can inhibit viral replication. It's been proposed, for instance, that the high fever, muscle aches, and pain that you experience from flu is due to the induction of interferon rather than virus replication per se. There's stimulation of the immune response that leads to more effective and faster clearance of viral infection. But probably most importantly, it also leads to the induction of the antiviral state. So let's look at what that is. Okay, so with the antiviral state, viruses will fail to replicate, or they'll replicate to much lower titers. The antiviral state is multifaceted, and interference with the replication of viruses may occur at different stages of their replication cycle. The effect depends on the virus and the host cell. In particular, what we see is interference with translation of specific viral messenger RNAs. There are two primary pathways here, and both require double-stranded RNA to be present. So one pathway is the 2,5 oligo-A synthetase. This synthetase is induced by IFN, but requires double-stranded RNA as a cofactor. The 2,5-A is, in turn, a cofactor for a latent ribonuclease, RNase L. RNase cleaves single-stranded messenger RNAs. Oligo-A is easily hydrolyzed, so activation of RNase L is transient. PKR is a protein kinase. Normally present in low levels, by concentration, it's increased as a result of interferon induction. It also requires double-stranded RNA. On binding double-stranded RNA, PKR is autophosphorylated, causing it to be activated. Once activated, it phosphorylates the translation initiation factor, EIF2 alpha. When this happens, the initiation factor cannot be recycled and protein synthesis is shut down. The effect of IFN is to prepare the cell for instant shutdown if it becomes infected, while leaving uninfected cells alone. So this is what you see. Up at the top left, you have a cell becoming infected with virus. This triggers the production of interferon. The interferon is released. Now remember, interferon is not going to help that cell that just got infected. Interferon is a cytokine. It's a signaling molecule. It's going to have its effect on other cells. So the interferon is released from the infected cell. It binds to the appropriate interferon receptor on an uninfected cell, and then it creates the antiviral state. It's signaled to that cell to be prepared in case a virus should infect. And then what can happen is what you see at the bottom. A virus comes along and double-stranded RNA can trigger the 2,5-A pathway that can lead to messenger RNA degradation, or it can trigger the PKR pathway leading to ribosome modification. It's significant that double-stranded RNA will trigger this because that is a very good indicator that a cell is infected with virus, because normal cells do not have double-stranded RNA. Think about that for a minute. We have double-stranded DNA, but not double-stranded RNA. Double-stranded RNA is a molecule that forms as a result of RNA virus replication. So the fact that double-stranded RNA is triggering this event is significant. Interferon was discovered more than 50 years ago. Initially, there was great hope that it would lead to treatment for viral infections. In general, however, it's been disappointing. Interferon alpha has been used to treat viral infections including chronic hepatitis C, hairy cell leukemia, Kaposi's sarcoma caused by HIV, and chronic myelogenous leukemia, or CML. It's also been used against lymphoma and malignant melanoma and warts. But interferons aren't used as often to treat most of these diseases anymore. Newer drugs have come along that work better and faster. One new thing, though, is that inhaled interferon beta is being studied to see if it can help treat COVID-19 infections. Now, interferon is used for the treatment of human papillomavirus. HPV is a common condition that often manifests as warts in various parts of the body. There are many ways to treat warts, including the use of immune response modulating drugs such as interferons. Interferon alpha is injected into the base of the wart using a small needle. This is done for each wart 3 times a week for 3 weeks. Since there are no pills available for this drug, and unlike treatment for other disorders, it's not given systemically. A less common form of interferon is available in a cream, which may be applied directly on the skin lesion. RNA interference is a natural process with a role in the regulation of protein synthesis and in immunity. It's also a potent tool for the exploration and manipulation of gene expression. In response to double-stranded RNA, cells trigger a 2-step reaction. In the first step, long double-stranded RNA is processed by an enzyme called dicer into small interfering RNAs, or siRNAs. These subsequently serve as the sequence determinants of the RNAi pathway by directing cleavage of homologous messenger RNA via an RNA-induced silencing complex called RISC. This is an ancient mechanism for protecting the host, protects its genome against viruses and rogue genetic elements, and has been observed in essentially all eukaryotic cells. Now we want to switch over to the adaptive immune system. Lymphocytes are the key cells in the adaptive immune system, particularly the B cells and the T cells. It generates an efficient and selective immune response, and there are 2 types, humoral and cell-mediated. Humoral immunity is also called antibody-mediated immunity. With assistance from helper T cells, B cells will differentiate into plasma B cells that can produce antibodies against a specific antigen. The humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. Antibodies produced by the B cells will bind to antigens, neutralizing them, or causing lysis or phagocytosis. Cellular immunity occurs inside infected cells and is mediated by T lymphocytes. The pathogen's antigens are expressed on the cell surface or on an antigen-presenting cell. Helper T cells release cytokines that help activated T cells bind to the infected cell's MHC antigen complex and differentiate the T cell into a cytotoxic T cell. The infected cell then undergoes lysis. Here we can look at how antibodies are produced. and it's through the clonal selection of a B cell. B cells are essentially antibody factories. They're made in bone marrow. When the B cell leaves the bone marrow, it has protein molecules called B cell receptors or antigen receptors on its surface. If the receptor molecule on the surface of a B cell happens to encounter an antigen to which it can bind, the cell is selected to proliferate. This results in a clone of B cells with identical receptors that can bind to the same antigen. Some of the B cells are committed to antibody production, that is, become a plasma cell. These cells can crank out thousands of antibody molecules per second. But some B cells become memory cells. They are set aside to protect us against a subsequent attack by the same virus. If there is a reinfection and binding occurs between antigen and memory cell, they proliferate and begin to produce antibodies to defend against that virus. Here we can look at a chart showing how immunological memory works. The selective proliferation and differentiation of lymphocytes that occurs the first time the body is exposed to an antigen is the primary immune response. About 10 to 17 days are required from initial exposure to antigens for the selected lymphocytes to generate the maximum effector cell response. If the individual is exposed to the same antigen at some later time, the response is much faster and of greater magnitude and more prolonged. This is the secondary immune response. The immune system's capacity to generate secondary immune responses is called immunological memory. So that's what we see in this figure. At the bottom, we're just looking at time, and then we're looking at antibody concentration along the y-axis. And just look at the blue line first. So there at time 0, that's when the host is exposed to antigen A. Now remember, the adaptive immune response takes a while. So the peak occurs sometime after 14 days. But then you'll see it rapidly declines. But remember, we have plasma cells and we have memory cells. The memory cells are still there. So if there's a second exposure to antigen, then you have the secondary immune response to antigen A, which is going to be higher and faster than what we saw the first time around. That's immunological memory. That is the basis, that's the science behind how vaccines work. That's why we don't get infected multiple times with the same virus, because of immunological memory. Now, if you look at this chart, look at the red line. That's antibodies to B. So at day 0, you're exposed to antigen A. Then 30 days out, you're exposed to antigen B. Notice that the shape of the graph in the response of antibody concentration to antigen B is the same as the original response was to antigen A, because antigen A and antigen B are not related, so the memory cells do not become involved in this reaction. The binding of antibodies to antigens to form antigen-antibody complexes is the basis of several antigen disposal mechanisms. And we can see some of these here. So first we have neutralization. The antibodies attached to molecules the virus must use to infect the cell. Therefore, the virus can't infect new cells. We talked about neutralization back when we were discussing methods of virology. Then we have agglutination. This results in the opsonization of the virus, and the large complexes are easily phagocytized by macrophages. Then there's precipitation. You have cross-linking of soluble antigen molecules to immobile precipitates. Or you can have activation of the complement system. When we look at neutralization, agglutination, and precipitation, they all enhance phagocytosis, so the removal of the pathogen through macrophages, scavenger cells. But complement fixation, that causes cell lysis. And again, remember, we've talked about complement before, but let's look at the complement pathway as part of the adaptive immune response. Okay, so let's look at the complement pathway. So you have an infected cell over at 1, and it's expressing antigens that antibodies recognize. You have antibodies attaching to these antigens on the virus-infected cell. Complement proteins can come along and link two antibody molecules. When this happens, the complement proteins are activated, and they release those proteins are released, and they attach to the membrane in a step-by-step sequence, forming a membrane attack complex, or MAC. The MAC forms pores in the membrane, which interferes with the physiology of the cell and ultimately causes cell lysis. Let's talk about the different kinds of antibodies that exist. When I use the term antibody, I think most of you are probably thinking of IgG, but there's other classes of antibodies that are important in controlling virus infections as well. So first, let's look at IgM. It's a pentamer. It's the antibody that's first produced by the B cells. It's large and shaped like a pentagon. This one is very good at agglutination. It's also very good at activating complement. IgG is what the B cells switch to later in virus infection. IgG readily crosses the walls of blood vessels and enters tissue fluids. It can cross the placenta and confirm passive immunity to the fetus. And then we have IgA. That prevents attachment of virus to the epithelial surface. It's found in body secretions such as saliva, perspiration, and tears. IgA is present in mother's milk. This is worth thinking about because, for instance, if you're testing antibodies to determine if somebody's been exposed to a virus, IgG antibodies are long-lasting, so they'll tell you if somebody has had an infection, but it won't tell you how long ago that infection was. If a patient is expressing IgM, or I should say is positive for IgM antibodies, that's an indication of a newer infection because those antibodies will disappear very rapidly. Cytotoxic T cells are a critical component to the cell-mediated immune response. An infected cell will display an antigen fragment on its surface using a class 1 MHC molecule. A specific T cell is activated by binding to the MHC antigen complex. The activated T cell discharges perforin molecules which will create pores in the membrane of the infected cell. This allows water and ions to flow into the infected cell and causes the cell to lyse. Viruses and their hosts have co-evolved for millions of years. During this coevolution, the hosts have equipped themselves with an elaborate immune system to defend themselves from the invading viruses and other pathogens. Viruses have evolved a diverse array of strategies to evade the host's immune responses. These strategies are as diverse as the viruses themselves. In general, each virus uses multiple strategies for immune evasion. We can divide the strategies into two types, avoidance and inhibition. With avoidance, the virus hides from the immune system but does not actively affect the host immune responses. For example, RNA viruses mainly rely on antigenic variability as the principal immune evasion mechanism. In latent infection, the full viral genome is retained in the host cell, but its expression is dramatically restricted, such that few viral antigens and no viral particles are produced. Immune privilege presents a special microenvironment where the systemic immune responses to allo and autoantigens are remarkably reduced. Immunoprivileged tissues include the pregnant uterus, eye, brain, and testes. These immunoprivileged tissues have critical, indispensable functions. On the flip side, immune privilege allows foreign invaders, including viruses, to find protection from the immune system within these tissues. Zika, Ebola, and HIV have all used the immune privilege of the testes to their advantage. In one of the most extreme cases, Zika virus was detected in the semen of a man in Italy at least 134 days after symptoms of the disease first emerged, even though blood and saliva samples showed no trace of the virus remaining. With inhibition, virus inhibits either the induction or the effector phase of the immune response. For instance, downregulation of MHC antigens on the surface of virus-infected cells is a strategy used by many diverse viruses. We see rapid shutdown of host macromolecular synthesis, interference with NK cell function. We'll look at a slide on that next. Inhibition of apoptosis, or some viruses directly infect T or B lymphocytes. Viruses have evolved mechanisms to avoid the host immune system, including means of escaping detection by both the innate and adaptive immune responses. Natural killer, or NK cells, are a central component of the innate immune system and are crucial in defense against certain viruses. To attain a state of chronic infection, some successful viruses have developed specific mechanisms to evade detection by and activation of NK cells. These NK cell-specific evasion mechanisms fall into two distinct mechanistic categories used in numerous virus families. But if you look at A, you can see that NK cells can be inhibited by a viral MHC class I homolog. In B, viruses can inhibit expression of HLA-A and HLA-B, which are human MHC class I surface receptors, and inhibit NK cells through the class I inhibitory receptors. In C, virus-encoded proteins can function as cytokine-binding proteins that block the action of NK cell activating cytokines. In D, NK cell activities can also be avoided by decreased expression of NK cell activating ligands in virus-infected target cells. And at E, viruses can also directly inhibit NK cells by infecting them or using envelope proteins to ligate NK cell inhibitory responses. The proteins in this figure that are in red are proteins that are virally encoded. Now remember, we talked about how type 1 interferons are a group of antiviral cytokines that are induced during viral infection by viral replication products, such as double-stranded RNA. IFNs exert their biological functions by binding to specific cell surface receptors. In turn, this triggers the intracellular IFN signaling pathway, mainly the JAK-STAT pathway, which eventually induces the expression of a large number of IFN-stimulated genes, or ISGs. The ISGs, remember the workhorses of the interferon response, set up an antiviral, anti-proliferative, and immunoregulatory state in the host cells. However, most, if not all, viruses have evolved a broad spectrum of strategies to block and interfere with the interferon pathway. Common viral strategies include A. Blocking of the IFN induction or expression, B. Intercepting receptor binding of IFNs through viral decoy IFN receptors, or C. Perturbation of the intracellular IFN signaling pathway, and D. Directly down-regulating the level of expression of ISGs. Viruses cause epidemics on all major cultures of agronomic importance, representing a serious threat to global food security. As strict intracellular pathogens, they cannot be controlled chemically, and prophylactic measures consist mainly in the destruction of infected plants and excessive pesticide applications to limit the population of vector organisms. So when we think about plant defenses, their defense against an initial invasion is using passive defenses, such as physical and chemical barriers, so essentially an innate immune system. When we look at physical barriers, things like cuticle, callus deposition, and so on, we'll look at that on the next slide. The chemical barriers, those tend to be directed against herbivores, including various kinds of insects that transmit viruses to plants. So plants, unlike mammals, lack mobile defender cells and a somatic adaptive immune system. Instead, they rely on the innate immunity of each cell and on systemic signals emanating from infection sites. If we look at some of the barriers here, you can see that lignin is a strong type of molecule that provides plants with a defensive structure similar to that of fibrous proteins. It acts as a barrier and can be found in wood and is characteristically found in plants that have recently endured pathogen attack. Lignin is the primary component of wood, and cell walls that become lignified are highly impermeable to pathogens and difficult for small insects to chew. Callus seals off sieve plates in the plant, effectively shutting off the transport of molecules around the organism. This is done to minimize the chance of the plant transporting infectious material around its own self and halting the movement of materials that could be used by the pathogen in aid of replicating itself. Ethylene promotes leaf abscission and is done to sever the plant of dead or dying plant matter. This is done to prevent the spread of infected material, therefore sacrificing infected sections of the plant, which is more economical than taking the risk of the infection spreading. Galls and tannins are created by the plant to encapsulate foreign agents found within the plant. A gall is an instance where an infected cell becomes inflamed that contains tannins. These tannins play a protective role by segregating the foreign agent and its chemicals from the rest of the plant. Faced with viral attacks, plants defend themselves through several resistance layers that are complementary in terms of defense timing, that is, early or late infection steps, location, in the first infected leaf or in systemic tissues, and targeting the virus-derived molecules, the viral genome or the viral proteins. plants have developed two major strategies to counteract virus infections: resistance gene-mediated and RNA silencing-based mechanisms. In A, we see a dominant resistance that relies on the interaction between an A virulence factor and a specific R gene product, and is effective several days after the virus entry into the plant. The viral pathogen is confined to the infected and neighboring cells. With B, we see a recessive resistance that corresponds to the absence of appropriate host factors that are required for the virus cycle. This is a non-inducible resistance. It's passive and effective throughout the plant colonization. It confers resistance at the infection step that requires the cellular factor of interest. With C, we see RNA interference. Remember, we talked about that earlier. And RNA interference targets viral nucleic acids. Once set up, after a few days, the effectiveness of this defense mechanism increases and spreads to the whole plant through a relay amplification process. And in D, we see hormone-mediated resistance against viral pathogens. This is represented here by the role of salicylic acid and methyl salicylate in systemic acquired resistance. In the next lecture, we're going to look at pathogenesis from the aspects of dissemination, virulence, and epidemiology.